Hello, everybody, and welcome to our last lesson. It's all about meiosis, or some people say meiosis. I've always said meiosis. And there are some cutouts that you're going to need to use in the future here in a little bit. So really, this is about reproductive cells. How do sperm cells and egg cells form? There are two different types, right? You have somatic cells, which are the cells of the body. They are diploid, which means they have two copies of each chromosome. So if I took a sample of your hair, I could reconstruct you completely from that sample. Like I could recreate you completely. It has every copy of every chromosome. And so I could recreate you. 99% of the cells in your body are diploid cells. Like most of the cells in your body are diploid cells, right? And it makes sense, right? Like you want to make sure that you have a complete copy of everything so that if your cells get damaged, like let's say like, all of a sudden like you get, a paper cut, you wouldn't want your body to be like, okay, I need to replace that skin with liver. I need to replace that skin with some other random thing, right? You want it to match. Now remember, we talked about mitosis all the way back in Unit 5. Now, the way you make diploid or diploid cells is mitosis. You start with the same number of chromosomes, you end with the same number of chromosomes, right? What makes germ cells, and when we say germ, by the way, germ means seed. It doesn't mean like, ooh, gross bacteria. Those are found only in your reproductive tract. Sperm cells, egg cells. They are haploid. Hap means half. They only contain, every sperm and egg cell only contains one copy of each chromosome, not double. Why do you think that's important? Think about it, right? You can pause, think to yourself. Well, if the number of chromosomes were not halved, every single time your cells divided, we would gain chromosomes every generation. So that means that your children would have double the amount of chromosomes. Your grandchildren would have 4 times the amount of chromosomes as you. Your great-grandchildren would have 8 times as many chromosomes as you. And so it just would get out of control really quickly. Okay, let's go through it. What is it called when germ cells undergo that special type of cell division that cuts the number of chromosomes in half? That is called meiosis. Here are the steps. This is meiosis 1. This is meiosis 2. There are 2 sets for it. Now, in your notes, on the very last page of your notes, you have space for some drawings. You're going to need different colors. And you're going to want to draw something that looks like this. This is the nuclear envelope. Here are our 2 chromosomes. Use 2 different colors. These little green guys are centrioles. Remember when we talked about cell division, about centrioles, and what they do in animal cells? So they're right there. So draw this first. Pause it if you need to. This is meiosis 1, prophase 1, metaphase 1, anaphase 1, telophase 1. Now here's what the cutouts are for. I want you to attempt to match the definitions for meiosis 1, prophase 1, metaphase 1, anaphase 1, telophase 1, and cytokinesis. So cut them out. See before you like glue them down onto that page, that empty page, like make sure that like they're correct though. Let's go through them. Meiosis 1, the first half of meiosis that results in 2 cells. That should have been super easy. Next up, prophase 1. The first step, nuclear envelope breaks down. Homologous chromosomes pair up. What does that sound like? It sounds like prophase from mitosis. Metaphase 1, the second step, spindle fibers form. Homologous chromosomes pair up in the center of the cell side by side. Anaphase 1, spindle fibers rip apart the homologous chromosomes, moving them to opposite sides of the cell. Telophase 1 and cytokinesis, the fourth step, spindle fibers dissolve, nuclear envelope reforms, the cell pinches apart, forming 2 separate cells. Now we are going to draw that on that diagram at the very end. So in prophase 1, what is happening here? Your chromosomes are touching each other. They've now formed X's. They're no longer those little single lines. They've formed X's. Notice the nuclear envelope is disappearing. It's dissolving. Your centrioles are moving to the sides of the cell. Now in metaphase, they are lining up in the center. These are your spindle fibers. They're cutting right through the centrioles. Sorry, the centromeres. They're surrounding everything. When they get ripped apart in anaphase, now pieces of the previous chromosomes are now attached to opposites. So in my head, I kind of just took a little chunk and kind of threw it up there in the corner. And then notice this half is still the same, right? Now we've kind of moved and merged things together, but now they're ripping them apart. In telophase 1 and cytokinesis, same thing. These are disappearing now, but they're being dragged over. The cells are pinching them apart. Pause it again if you need to. Next up, what's super important? Why were they touching each other, right? Why? In prophase 1, crossing over happens. This is where we get all of our genetic uniqueness. This is why we are not a perfect 50-50 mix of one parent and the other parent. This is why you might look a little bit more like one parent than the other parent. This is where all of our genetic diversity really comes from. It comes from prophase 1. It's called crossing over. In telophase 1, 2 cells are produced that are genetically unique. These are now no longer the same. So if I took one of your sperm and egg cells I wouldn't be able to recreate you. Why? Because I'm missing half of your genetic material, maybe more than half of your genetic material. We just talked about how crossing over is important. You can't clone yourself from an egg cell or a sperm cell of yourself. You could only really get half that information. By the end, we have not made anything halved yet. So by telophase 1, we now made genetically unique things, but we haven't cut the number of chromosomes in half yet. All right, now, here's a little simulation of what crossing over looks like. They're crossing over, and now look, oh, little parts are different. Parts of the chromosomes have crossed over, and now they're different. Now we have unique differences in our cells at the end. Okay. I want you to do the exact same thing with your cutouts for meiosis 2, prophase 2, metaphase 2, anaphase 2, telophase 2, and cytokinesis. Match them, please. All right. Hopefully you got meiosis 2. It's now where you get 4 cells at the end. Okay. Prophase 2. The nuclear envelope breaks down again. Now spindle fibers form. And now chromosomes line up straight in the middle in a straight line. Now, our spindle fibers rip apart the sister chromatids, moving them to the opposite sides. 4th step, spindle fibers dissolve, nuclear envelope reforms, and now our cells pinch apart and form. It should say 4 separate cells, not 2. Okay. Here's what we got. Notice, nuclear envelope's going away, guys. We still have our X's. Now they're lined up in the middle. Again, our spindle fibers are surrounding them. Now here's where we split things in half. Notice I now have a little orange guy on this side, a mostly blue but kind of orange guy on this side. Blue guy on this side, mostly orange but also a little blue guy on this side. And then this is telophase 2 and cytokinesis. Nuclear envelopes reformed. Notice we now have genetically unique cells. They're all different from one another. Okay, what's important? When does all of this important stuff happen? In anaphase 2, this is where genetic information has been halved. In telophase 2, we end up with 4 cells that are genetically unique. Please add this key somewhere. Otherwise, you're not going to know what those dashes mean or anything. Spindle fibers are the little dashed green things. Centrioles are the dark green little circles. The nuclear envelope, that is this purple thing. These are our chromosomes. These are our homologous chromosomes, the X's. And that is the end of meiosis. Thank you very much. Again, if you have any questions, let me know. But the back of your notes should be completely filled in now.